Yeah, good afternoon. Yeah, welcome to this edition of the NITheCS Colloquium. Sorry, this is my laptop that is listening. Voila, sorry, the echo is gone, sorry. You would imagine you would have learned after 100 or so of this colloquium to switch off the volume. Anyway, so good afternoon. Welcome to this edition of the NITheCS Colloquium. My name is Francesco Petruccione, and now I'm the director of NITheCS, not anymore the interim one. In fact, I'm the substantive director of NITheCS, but maybe some of you can explain me what it means. Anyway, this afternoon, we have the big pleasure of having Professor Marlo Möller with us. Marlo is based in Stellenbosch, and now I read it because I don't want to say anything wrong. That's not me now. In the division of molecular biology, we are probably too close to each other with two microphones, and human genetics. And she's also associated to the Center for Bioinformatics and Computational Biology at Stellenbosch University, which is perfect because quantitative biology and bioinformatics is one of the teams of NITheCS. Yeah? So Marlo, we are very grateful that you're here with us this afternoon, and we are very looking forward to your talk. Thank you very much.
Thank you very much, and thank you for the invitation. I don't often come to Stellenbosch campus because we are at Tygerberg campus and in this beautiful new building, the Biomedical Research Institute that you can see on the slide. But I am happy to be here today and to tell you what we get up to on the other side of the world. So human genetics in the BMRI, we're a couple of groups that share the lab there. So our group is part of the infectious diseases group. Then we've got the Parkinson's disease group with Prof. Soraya Bardin, the rare diseases genomics group with Prof. Shahida Musa, and then the neuropsychiatric genetics diseases group with Prof. Sean Hemmings. So in our group, we've got Professor Craig Kinnear, you can see here at the bottom. There we go. Myself, Professor Desiree Peterson, Dr. Caitlin Uren, and Dr. Brigitte Glansman, and we're the senior researchers in the group. So we're mainly interested in the genetic epidemiology of TB, but we also take the work to a functional level where we study the effects of variants. We're interested in extreme phenotypes of disease and very much interested in population genetics. So I want to start with acknowledgements. Everything I show you today would not have been possible without our amazing study participants and people who generously give of their time and their blood and sometimes their saliva. And then I also want to thank our funders and very importantly, all my collaborators and colleagues, and you'll see their faces in the pictures as I go through the talk. So today, because we're such a diverse audience, but I see some geneticists in the audience, so sorry that I'm going to bore you, I thought I would start with some concepts in genetics. Then I'll move on to discuss genetic diversity and why it matters, and I'll discuss several examples, and I hope to convince you that it's really important to study and include Africans in these studies. And then I'll conclude. So I had this quote from someone who said, I always thought that geneticists were really clever until I realized that they only work with 4 letters. And that's a very simplification, but it is true. So we study A, T, C, and G, and your DNA is everywhere. It's in your skin, your saliva, and even in your hair. And you can think about your DNA as a book. And every page in this book is a gene. And your genes give the instructions to make you, and you inherited this from your parents, which is why you look like them. So Gregor Mendel was the father of genetics, and he turned 200 in 2022. And I was looking for a better example, but his classic experiments is still the best out there. And if you studied any genetics course, you would have seen this. So he was interested in the characteristics of pea plants. And this is just a perfect example to distinguish between genotype, phenotype, and alleles. So an allele is a version of a gene. And basically, this is what you inherit, the heritable unit. So Mendel studied this gene that determined the color of the flowers. And the small allele was for the white, and the capital letter was for the purple. So if you look at the allele, you expect that each pea plant will carry 2 copies. So it can then either be purple with 2 capitals, purple with a capital W and a small one, or white with 2 little alleles. And this is then the genotype frequency. In other words, how often do we see each allele combo? In terms of phenotype, this is how often we see white versus purple, so what you see walking around. And then the allele, as I say, is just how often we see each allele. And this is all that you need to know to understand genetics. So there's very different types of genetic variation. And today I'm only going to speak about single nucleotide polymorphisms. They're the most simplest form of genetic variation. And what you'll see is that it's a single base pair change in your DNA. And this can affect your phenotype as you change. So SNPs can generate biological variation because it changes how the gene is then transcribed. So some SNPs can change how you look, how you respond to disease, how you respond to treatment, and then some lead to no changes at all. So a simple example of this in humans is the ABO blood system, where your parents can be A positive or have an A blood type or a B blood type, but they carry the recessive O allele. And then their children can either be A, AB, B, or O. And we've seen enormous technological advances in SNP detection. So when I started my PhD, we were still genotyping single variants laboriously in the lab, and it took you 2 years just to optimize this. Then the technology advanced and we were able to buy microarrays. So suddenly you could look at hundreds of thousands and millions of SNPs. And then sequencing progressed so much that we could actually look at the whole genome and also at all the protein coding genes, which is the exome. But we had to break up the genome in small pieces and then paste it back together to study. And now the technology has advanced to the stage that we have long-read sequencing, so we can sequence the DNA without breaking it up, and it's easier to align and work with. So I also wanted to point out today that all humans are 99.9% similar, and there's only 0.1% difference between us in our genes. So humans are also 60% similar to the chicken, 84% to the dog, 92% to the mouse, and 98% to the pig. So if you look at some people and how they drive on the roads, this is not hard to imagine. But what I want to say with this is that there's only one human race. We are not subgroups or sub-distinguished. And the 3 billion base pairs in our genomes will have changes, yes, 0.1%, which is a couple of million of changes. But it demonstrates that you can't subcategorize humans, and we're not biologically distinct. It's just migration patterns are what you were exposed to that influence your DNA. And to further explain this is that you can see that human genetic variation is actually distributed around a gradient. So there is considerable genetic overlap between populations. So this is a principal component analysis in the first two principal components. And the colored dots are reference populations that were sampled from continental areas. And you can see that the spread is based dependent on how they were sampled. The gray dots are from a biobank, a diverse biobank in New York called BioMe. And here you can see the diversity spread across the regions. So again, anyone that tries to rank humans based on human genetics, they fundamentally don't understand what genetics is about. And I always put this up because I feel like I can't talk about the research participants and not disclose my own ancestry. So geneticists just use the term ancestry to characterize the genetic origins of an individual or of a specific population. And what we actually simply mean is there is a difference in allele frequency. It's nothing more complex than that. And this is also an example where phenotype doesn't always match genotype. And then I always throw this in as well, because movies like Gattaca from the 90s try to convince us that your genes is your destiny, and this is what's going to happen to you, even though the lesson in the movie is a bit different, but I encourage you to go watch it. But instead, your DNA is just a box of tools and materials for the true architect of life, the cell. So there's an intricate play between many factors. This can be how your RNA is transcribed, the epigenetics of your genome, and also the position of the gene in your genome, together with environmental factors that you are exposed to. And then this is one of my favorite slides because it shows how humans migrated out of Africa and to populate the world. So what we show here is that when people migrated out of the continent, the genetic diversity decreased as we moved out of the continent. And this is the basis of our study in Africa, of genetics in Africa. So the gap between European ancestry databases and those of everyone else has been highlighted multiple times by very famous geneticists and all over the world. But despite all of this and many commentaries and initiatives, we can see that this gap between Europeans and the rest of the world is still there and still growing. So this is a graph called the GWAS Diversity Monitor. It's a website that's live. And you can see how over the years, the European studies always dominated. And yes, here you can see some Asian studies coming through, but you can barely see the African studies all from Latin America and other countries. So then the question is, does this gap really matter? And that's what I hope to convince you of from today. So this is a really simple example. This is the so-called Duffy allele frequency. And if you look at this and you see all the FYO allele, this is the green one in the African continent, you might think that this allele is not there and that it was selected for with Neanderthal admixture when people migrated out of the continent. So you can see then the FYA and the FYB allele in the rest of the world. But then when you include two South African populations, the Kumani San and the Zulu population, you see that the FYA and FYB allele were actually present here. And we've dated it, it's been dated back to be about 60,000 years old, and it's been segregating therefore in Africa for this time. And then when populations moved into the equatorial region, it seemed to have been selected for. And this allele is associated with resistance to malaria, which at the time was prevalent in Africa, but is not anymore. So the Duffy allele is then correlated with the distribution of malaria at the moment. So the idea is that there was an ancestral pathogen that was Plasmodium-like, and that caused the selection for this Duffy allele in the equatorial regions. And this was about 40,000 years ago. And then when humans moved out of the continent, the allele appeared again. The second example is the Human Genetic Diversity Panel. So this is a very old study, but it was a great study at the time because they actually included populations from all over the world. And what they did was they divided it into 7 colors. So basically, this was to show that there is genetic diversity across the world, but they chose the subdivisions. And basically, here they chose 7. So Africa, Middle East, Europe, Asia, East Asia, and then Oceania and America. And this can then be misinterpreted as supporting this racial categorization. But it's not the case. And it doesn't really show where the diversity is high. And what was also concerning about this study was that 85% of the sampling was outside of Africa and 15% was inside of Africa. So a follow-up study then asked, what if we flip this around and sampled 85% within Africa and 15% out of Africa? And at this stage, more genetic data became available. So what you could see here is that there's actually very distinct ancestries in Africa. Yes, the Nama population from South Africa, the Zulu, they've got their own distinct ancestry, and then the West African, and so you can go on. So the point here is that there's no such thing as an African ancestry. We're very much more diverse than this. And because we've been undersampling diversity in Africa, we've always left them out. And if you don't include them, you get a strikingly different picture from what you would have gotten if you included them. So what makes South Africa special in this context? Well, we're a hotspot for genetic diversity. Not only do we have African ancestry, click-speaking hunter-gatherers, Asian and European ancestors, because everyone came together here at some point again. And this led to a large degree of genetic diversity. We also have unique environments and selection pressures. And this is evident in my third example, which was Mabry syndrome. It's a rare disease that is characterized by developmental delays, intellectual disabilities, seizures, and then also elevated serum alkaline phosphatase. And the clinicians saw these 3 patients and they thought this is definitely Mabry syndrome. So they sent them for genotyping at a commercial company and no variant was identified. They then sent the samples to us and we did whole genome whole exome sequencing and identified a novel variant. So what was lovely about this is that we then found this variant in 2 unrelated other cases. And all these individuals were from the same ethnic group. There was no apparent relatedness. So we think it's a bottleneck effect, that something happened in South Africa at the stage that selected for this variant. So it is more common in the population than we expected. And what makes this special is that Prof. Shahida Musa is a genetic clinician and one of the few in the country, was able to get this established as a genetic test at the NHLS at UCT. And it's important because then you can do accurate genetic counseling, prenatal testing, and treatment. And she then discovered 2 patients who didn't present with this high ALP level that is usually characteristic of Mabry syndrome. Instead, they had low levels. But because of the genetic test, she was able to accurately diagnose them instantly. And then another interesting feature of Mabry syndrome is that this patients, when they get treatment for their seizures, it doesn't respond to the standard treatments. But if they're diagnosed in time, it can be supplemented with vitamin B6, and then they respond to the medications. So this is a really cheap fix that can help prevent brain damage in these individuals. So the 4th example is a study that we started during COVID, and this was in children that suddenly presented with hyperinflammatory disease about 4 to 6 weeks after exposure to SARS-CoV-2. We did whole genome sequencing of them, and this is the only study in Africa investigating it, and did the alignment and characterized the variants. So we were able to show that MIS-C has a complex genetic etiology in South Africa, and we identified 38 possible candidate variants that has not been reported elsewhere. This is also the largest study of MIS-C patients in the world, but we are struggling to get it published, believe it or not, even though there are much smaller studies out there from the European side of the world. So why was this study important? Well, we can find novel susceptibility variants in Africans, and this can have implications for future pandemics, and we can reveal the cause of these severe phenotypes in Africa. It can also help us revise vaccination strategies and treatment strategies. And then, as I said, this is the first study in Africa, and it might be the cause of this disease might be a bit different in Africa. So we've since expanded the study to Kenya, and that's a wonderful data set that is also going to be published. So the next example, and this is my main focus of research, is tuberculosis genetic susceptibility. So TB is still a big problem, and we're not investing enough money in it to treat it, but also to prevent it. And we actually only have one vaccine that works only in children and not in adults. But infection with Mycobacterium tuberculosis is not sufficient to cause disease in most humans, and 90 to 95% of individuals who are infected will not develop active TB. But we are interested to find out why some of these do. And the way we do it these days is with genome-wide association study. So basically, we genotype millions of SNPs on the microarrays, and then we compare it between our cases and our controls. But as opposed to other phenotypes with TB, we didn't find much overlap in these studies. So there's been some really large studies done in the world, but the overlap here is really terrible. So we set out and we joined the International TB Host Genetics Consortium, and this was 12 cohorts with about 15,000 TB cases and 17,000 healthy controls. And again, we divided up in this simplified continental ancestry. But we found a significant association in the HLA class 2 region. And this is a highly polymorphic gene on the chromosome 6. And this is just a plot that shows where we found it on the chromosome. And this paper was published in eLife. So HLA class 2 is a key determinant in the immune response to TB. And MTB has mechanisms that directly interfere with MH class 2 antigen presentation. So I want you to remember this name because I'm going to come back to it. So our previous population genetics work showed that the populations in the Northern Cape is very genetically diverse. And some of these populations have lived in Southern Africa for more than 100,000 years. And historically, these populations were larger and therefore contained a lot of genetic diversity. So while we were doing our population genetics work, we noticed that there was also a very high incidence of TB, and this resulted in the formation of the NCTB study. So to date, we've collected about 2,500 individuals. The Northern Cape is the largest province in South Africa, but the least populated. And it has a lower HIV rate, but high TB. So normally increased HIV rates is also associated with increased TB. And then we did our sampling at community health clinics in the area and trained community-based RAs to help with sampling in the region. So we were able to create jobs for 7 people there. And Caitlin's in the audience today. She did some of the training. And here's some of the packets of saliva that we collected from the people. So what we're collecting here is patients who come to the clinic and who then test gene expert positive, in other words, positive for TB, but we also enroll people who had past TB episodes. And then the controls are people who test negative. And then we collect a whole bunch of demographic and behavioral data, including the weight of people. So this resulted in this paper where we could see what was the epidemiological correlates associated with overweight in the area. And at the same time, we're doing a study which we just call the diplomic study, where we collect participants from the general public, and we perform diagnostic tests to see if they were infected with latent TB infection. We also collect demographic data, and then we're going to do low coverage whole genome sequencing. So the idea is then to investigate the genetic factors that may predispose individuals to convert from latent TB infection to active TB. And as part of this study, we also have an outreach and training in the Northern Cape. And if you later have time, you're welcome to go watch this lovely YouTube video where we show what we did. So we visited schools and taught them about genetics. And we also gave some extra training to our community assistants while we were there. So our preliminary results from this study, we were able to genotype 362 cases and 482 controls at the time. And we were able to show that the TB risk was the highest in the small towns. We also showed that males are 3 times more likely to progress to TB than females. And we were genotyped 182 individuals just to look at the ancestry. And as you can see, it's predominantly Khoisan ancestry, which means we can expect a lot of genetic diversity. And to analyze these populations, we need new tools and new approaches. So we've looked at admixture, we've tested different programs in these populations, we've done a de novo assembly of a South African genome, and we've looked at local ancestry inference in the community. So local ancestry is basically just looking at the specific position in the genome and determine which ancestry this region was inherited from. So these individuals are all from the same community, but you can see they have very different ancestries. And when you incorporate this in an association study, you might introduce confounding. So we've incorporated this in our TB genetics association studies, and we were able to identify ancestry-specific disease risk alleles that were associated with TB and that we previously missed in our analyses. So not only did we have updated computational algorithms to do the analysis, but we had better genotyping platforms and we had better reference data sets. And we were able to identify the TERAP gene that's involved in toll-like receptor signaling, and it recognizes microbial pathogens. But more importantly, we were able to confirm the HLA class 2 association that the International TB Host Genetics Consortium found. So this paper is currently under review, and here were the 4 models that we tested. So you can see with all the other models, we didn't find any genome-wide significance, which is a solid line there. And here was a less stringent significance. So we almost reached that when we corrected for local ancestry. So if we had a larger sample size, we expect that we would have reached this. Another example of where we incorporate ancestry was in a EQTL study. So this is where we combine genotyping data with RNA sequencing data. And when we analyze these genes together, we could see that our association signals overlap with previously reported association signals. And this is very important because, as I showed you earlier, replication in the TB host genetics field is really hard to find. We're also busy with a study looking at pharmacogenetics. So what people have seen is that They've studied pharmacogenetics and kinetics in Africa, but they don't find the same variants that people reported in European studies. So this study is funded by Novartis and GSK, and we are collecting these samples and we're going to genotype and sequence them to find the African variations that contribute to the way that you metabolize drugs. We're also training students, and the idea is then to have these combined data sets available for additional analyses at the end. And in the end, we hope to have matched genetic and PK data of 1,500 individuals. So this is my last example. And despite broad agreement that that Homo sapiens originated in Africa, there's considerable uncertainty surrounding the models of divergence and migration also across the continent. And then our progress is also hampered by a shortage in fossil and genomic data. So this is a basic model that would describe genetic diversity. And again, you can see that we broke it down it was broken down into the Yoruba, the Europeans, and then Asian groups. And there was more focus on what happened after the bottleneck when people left Africa. So here, this population size was left constant for the Yoruba. And after the bottleneck, they thought that it was about 2,000 people left, but even fewer. So you can see there was not a big focus on what was happening in Africa at the time. And this was the only way to do it. We had to simplify things at that stage because there was not enough genetic data available. And this is just what people did about 15 years ago. So 10 years later, there were many more genetic data sets available, and people started to think a bit differently about how these models might work. So at the time, there were two models that were mainly discussed. The one was the single origin model, or the recent expansion. And there you can see the three different regions. So from South Africa, that then split off to West Africa, East Africa, and Europe, and then the rest of the world. And then that was a single recent expansion, basically from a Southern African origin. And as an alternative, people had also approached archaic admixture. So this was when people found evidence of Neanderthal and Denisovian admixture in Europe. And so they said, well, there must have been other species living in Africa at the time. So we proposed this model, and that there was some admixture in the Western Africans and going on from there. And this was actually shown to be true with genetic data. But this is two very simple models. So then there are some more complex models that were proposed, and that's that there was many different populations living in the ancestral stem within Africa, and that some of them also contributed to the contemporary African populations, but they're not archaic. In other words, they were just other modern humans that were living within Africa. And then people also proposed what is called the Africa multi-regional model. And this was just that the divergences among the regional groups go back 300, 400, or 500,000 years. So this is a very deep divergence compared to the rest of the studies. And it's very deep compared to the bottleneck that was about 50,000 years ago. So these are the four models that paleoanthropologists proposed. But the question was to test these models using DNA from contemporary people. And what we had here was that we were able to get real DNA from Africa from several different sites, and we could model 6 populations simultaneously from these regions. We also included the Neanderthal genome in this. So the key here was to look not only take the West African populations, but to look at the diversity on the continent. And in particular, I want to highlight that we published the largest NAMA genome data collection to date. And this allowed us to use 380 genomes that we then used to make this new model. And we also developed a new computational model called MOMENTS, which used linkage disequilibrium between the populations. So the study started by using the 4 different models that I just told you about, and we just tested which one worked the best. So amongst the models that we found, this one actually performed the best, and it's called the merger model. So here we said there's actually no archaic admixture. We're not seeing some African species that admixed with humans in Africa. But there was very deep divergent regions in there. So you will see stem 1 and stem 2. They coexisted and they all had gene flow between them, and that's what the arrow mean. And you can see that this was about 1 million years ago that they split into two. So there's stem 1 and 2, and they are exchanging migrants. And then something happens, and we see the structure in the population. And we see that there's a split between stem 1s and stem 1e. And now these populations are separate. So we expect that this is at a time when Africa was really cold and people weren't migrating all over the continent. They were stuck where they were at the time. And this just means that the stems then fractured further. So then 120 years ago, what we see is that these populations started to come together again. And this is where we see the regional populations that we recognize today. So here you can see that stem 1s and stem 1e merge again, and they contribute to what we know today as the Nama population. And then stem 1e here merges with stem 2, and this turns into the West Africans as we know them today, and they then split off into the rest of everyone else and also those outside of Africa. So it's quite a complicated structure, and then we see that people merged together later on. So what's interesting is that there's this regional persistence across the different groups that we still see until today. So in conclusion, the power of contemporary African DNA lies in its genetic diversity, and there's still extraordinary opportunities for human genetics research in Africa. It can help us to decipher disease and response to treatment, and it can also inform evolutionary studies and population genetics. So it shows the amazing complexity of local adaptation and migration. But we still require more funding to overcome the current lack of genetic representation from the continent. Thank you very much.
Very much for the fascinating talk. We have plenty of time of questions. Any questions from the audience here or from the audience online? No volunteers. Donovan, are any questions online? Not yet.
I see a hand.
Here we go. Sorry? Then maybe Donovan, can you give the permission to speak to Andy Moore? Andy, you're welcome to ask your question.
Thanks very much. This is a non-geneticist speaking, so it might be a very dumb question, but there was an idea that anatomically modern humans, if you looked at the DNA, the mitochondrial DNA, that they coalesced to a common ancestor around about 200,000. And this was interpreted to represent a major population decline. Does that still fit into your model, or is that now rejected?
No, I think we are looking at time periods before that, so it still fits into our model.
So does that mean that you know, if you go back, they're the Moroccan fossils that have been dated at 300,000, which have been suggested to be modern humans. But would the genetic bottleneck then be later than that, at 200,000, so that there would have been an expansion from that small population, or has that idea changed?
So I think it has changed. Let me see if I can just go back to our model again. It's stuck here. So it's just more complicated than we all imagined, right? I think everyone's trying to show that humans originated in this one place in Africa and then spread, but it's not as simple as that. There were multiple interactions. I don't know if you can see my pointer online. So you're talking about 200,000 years ago, which is here. So you can see at that time, we're saying people were quite separate in separate areas in the continent. And then about 120,000 years ago, we came together again.
Okay, so the stem one would have been Nama, is that correct? Or
No,
gave
stem
rise
one-
to the Nama, and step two would have been East Africa. Stem two, sorry, stem one.
So stem 1S gave rise to the Nama, and then stem 2 was West Africa, and then stem 1E contributed to East Africa.
Okay. No, thanks very much.
Question? Are there any more questions? Over there. Up there.
Hello? Okay, it's a bit of a weird question, but so people migrated over Asia and then towards the Americas. If South America were isolated for long enough, would people have evolved enough so that we wouldn't be able to reproduce with a new species of human? Would that have been possible? And if yes, how long would they have had to be isolated for that to happen?
That is an interesting question. So I would imagine there would have to be a very strong selection pressure for humans to not be compatible anymore to reproduce, so something like a huge climate change in the area, something that really had a strong selection effect. The time, I am not sure. As I say, it would have depended on the strength of the selection. I've never answered a question like that. Thank you. That's quite interesting thinking.
Yeah.
Okay, it's a question from David Scarf. Has any work been done to figure out how khoisan resistance to any diseases can be used to develop vaccines in other populations?
Nothing that I know of. And I think, given the current situation of the khoisan today, it would be very hard to do a study like that without getting proper consent from them. I'm also not aware of any phenotype where they have specific resistance to disease. We work with the communities there regularly and nothing has been reported.
You really want me to do the 10,000 steps?
Ma'am, do you think maybe the khoisan resistance to diseases and stuff originated from their original use of traditional herbs and medicinal weapons?
That is definitely possible. I know there's huge studies just going on about traditional medicine in the area. They even established a factory in the area where we work, where they collected the hoodias. So yeah, definitely there might be some medicinal reasons for people seeming to be more resistant. But as I said, in today, we don't notice that there's anything happening. In fact, we have seen a rise of cancer in the communities. So that might very well be environmental.
If there are no more questions, sorry. Okay, here we go.
Okay, Eckhart Schumann says, the coming together 120,000 years ago would have corresponded to the last interglacial. This would have meant a much greater mixing possibility, with a question mark.
I think so, yes. I am but a human geneticist, so my anthropological knowledge isn't that great. But yes, that would make sense to me. As I said, the hypothesis is that there were really cold areas in Africa at that time, and that people persisted in regions where circumstances was favorable.
Okay, then if there are no more questions,